And I guess another general question, could you give a brief comparison between embryonic stem cells and induced pluripotent stem cells, and then kind of the challenge that both are bringing to the table, whether ethically or ability to redifferentiate?
Yeah. So embryonic stem cells, so they're both pluripotent stem cells. Embryonic stem cells are derived from an embryo, in this case, a pre-implantation embryo. And those are, and so that's what those are, where, and as opposed to induced pluripotent stem cells, which are generally derived by reprogramming from a somatic cell type, maybe from an adult or maybe a neonatal cellular phenotype. They're very similar, but they're not necessarily identical. And a lot of that comes, the thought is that the embryonic stem cells are sort of our type specimen. And so for an induced pluripotent stem cell to be called truly pluripotent, it should have as many of the characteristics of an embryonic stem cell as possible. And a lot of that is technical. So depending on how you make the induced pluripotent stem cells, the procedures can either get them very, very close to an embryonic stem cell phenotype or a little further. They never actually completely replicate all of the genetic and genomic and epigenetic features of an embryonic stem cell, but they get very close. So obviously, the iPS cells are ethically a little easier to, conceivably utilize because they're coming from an adult cell type or a newborn cell type. So you're not destroying the individual when you create that cell, or you're not destroying the conceptus. Whereas embryonic stem cells, you do. And that obviously is much more of an issue ethically and legally and politically for human embryonic stem cells. It's probably less of an issue for animal embryonic stem cells. So this is why a lot of the iPS cell interest is focused a lot on the human because of the ethical dilemmas associated with human embryonic stem cells.
Thank you.